Surgery for brain cancer remains the mainstay of therapy and one of the main modulators of prognosis. But brain cancer is nearly impossible to cure with only brain surgery. Our team here at the Mayo Clinic, in collaboration with Purdue University, led research efforts that brought a new and exciting technology platform to the operating room capable of diagnosing brain cancer types in real-time patients with brain cancer, opening doors so one day we can bring the fight against brain cancer into the operating room with therapies beyond brain surgery. Diogo, can you tell us how we did this?
Absolutely. So we developed a workflow based on desorption electrospray ionization mass spectrometry, big word, so we call it DESI, where smears are made right outside of the OR using freshly obtained tissue and are analyzed in real time while the surgery is still ongoing. And we chose to focus on a unique biomarker, 2-HG3-hydroxyglutarate, which enables us to know whether the tumor is IDH mutant or wild type. So, Darky, could you tell us a little bit more about this technique, DESI, and how impactful it was for our study?
Thank you, Diogo. This is a mass spectrometry technique that uses a charged solvent that extracts analytes from a surface and send them to a mass analyzer. With this imaging, we can obtain chemical information from the tissue sections as well as tissue smears and analyze lipids or specific metabolites such as 2-HG. In this work, we focus on 2-HG given its unique quality in brain cancers. Our work is highly relevant given that nearly half of gliomas are IDH mutant, and there is a recent evidence that this can be targeted by specific inhibitors. Plus, knowing whether a sample has an IDH mutation or not allows us as surgeons to use that information as a surrogate of tumor infiltration. Our future goal is to achieve molecularly guided resections using this technology to maximize safe surgery until the borders are clear of any infiltration that can be detected with this platform. Diogo, tell us about our results and how many patients and from which countries were represented in this work.
Absolutely. So we included two completely blinded separate cohorts. In our cohort here at the Mayo Clinic, we analyzed over 240 biopsies from a total of 34 patients, so multiple biopsies per patient. That included margin, meaning the area of the infiltration of tumor into the normal brain, as well as core biopsies, that center of the cancer. And we found we were able to discern IDH mutation from wild type with 100% of accuracy when compared to what is the gold standard, which is pathology. We were also able to actually detect infiltration into those areas going to the margin in 88% of all the samples analyzed. And as I mentioned, we were able to replicate this in a completely different center in another country with also 100% of accuracy. So beyond our cohort here at the Mayo Clinic, which included actually patients from over 10 countries, that other independently run cohort from our international collaborator had over 100 patients, and they had the same results. So in total, when we put them all together, we've shown 100% accuracy over more than 350 biopsies in 140 patients. So, Darky, what do you think this means for our field in general?
Our results provide hope. Brain cancer is a disease that urgently needs better therapies, and I am excited and I am thrilled about the results we show in our intraoperative platform in this study in the proceedings of the National Academy of Science Journal. We demonstrate that we can bring the fight against brain cancer into the operating room with instruments that go beyond what we can do with brain surgery. It reminds us of what Dr. William Mayo said in 1935. It is a great thing to make scientific discoveries of rare value, but it is even greater to be willing to share these discoveries and to encourage other workers in the same field of scientific research. Thank you for your attention, and we look forward to many more discoveries here at the Mayo Clinic.